series from our research group focusing on drug-drug interactions, clinical decision support. The goal of our webinars and our research is to improve the relevance of drug-drug interaction warnings. Today we have a really interesting medication with respect to drug-drug interactions, and it's coming into favor in a couple different ways, and that's the drug colchicine, which is an alkaloid and has some interesting properties that we thought we would share with you. And when I say interesting, I mean negative properties that can result in life-threatening conditions. But before I do that, I would like to just thank the research team and show you who we are, representing different organizations around the country, and lots of different skills here, from informatics folks to clinical to drug interaction experts. So my name's Dan Malone, and I'm the principal investigator of this project, and I'll be your host for today's webinar. But I'm not going to be the primary speaker. I'm going to be turning it over to my colleagues. As a disclosure, conflicts of interest, majority of us have no conflicts of interest. Dr. Hansen and John Horn are publishers of the top 100 drug interactions, a guide to patient management. So they declare that conflict of interest. And this project is funded from a grant from the Agency for Healthcare Research and Quality, but the content is our responsibility and does not necessarily reflect the views of the agency. So just want to make sure that people know that. So if you've attended one of our webinars before, you've seen this particular slide. We know there's lots of defenses to prevent patients from receiving drug interactions, but when those defenses line up, or such that the holes line up, I should say, it's possible that a patient can experience an adverse drug reaction, harm from receiving two medications. And the medications that we want to focus on today primarily is colchicine, and it can be combined with a CYP3A4 or a PGP inhibitor. And then we have to start worrying about whether the patient's going to have adverse consequences. Now, I'll let Dr. Hanston talk about this flow diagram a little bit later, but the point being is colchicine is given in relatively small doses, between 0.6 to 1 milligrams, 1 to 2 milligrams, I should say. And it's done for a very good reason, and that is the medication is also a poison, more or less. And when it's present in the body in high levels, can result in all sorts of negative health consequences to the patient. So what I'd like to do is turn it over to Dr. Phil Hanston to kind of walk us through the clinical implications of this drug interaction and also the evidence for those drug interactions. So Phil, you want to take it from here?
Okay, thank you very much, Dan, and welcome to this webinar. I'm speaking to you from Santa Rosa, California, and this is the flower. It's a beautiful flower. The autumn crocus apparently has been used since Roman times for treatment of gout and related disorders. It's a beautiful flower, and it's effective, as Dan indicated, for a disparate number of different disorders, and new ones coming online recently. But the problem is, as he mentioned, it can be deadly if it's not used properly, as we will discuss. Okay, next slide. As I said, used for lots of different things, not just gout. It's used extensively for pericarditis and is very effective for that. Familial Mediterranean fever as well. It's one of the best medications for that. It's used for arrhythmias, a variety of dermatologic disorders, and about a year ago, a little over a year ago, a study out of Canada showing that it, in fact, was beneficial following myocardial infarction. So that might increase the use of colchicine substantially in the future. And there are, as we'll talk about at the end of this presentation, about 24 current trials of colchicine being used for COVID-19. Unfortunately, it has a lot of drug interactions and can produce sometimes untreatable toxicity. And the reason why I say untreatable is it's not dialyzable. And especially if the colchicine toxicity results in renal failure, which it can for various reasons, then you can't get rid of it. There's no way, and sometimes hepatic failure at the same time. And so the person just marinates in colchicine. The half-lives in people that are half-life of colchicine in people undergoing that kind of problem are sometimes weeks. So it's a very nasty thing to treat if it happens. Usually pancytopenia is the cause, rhabdomyolysis, and also, as I said, multi-organ failure. It's metabolized by 3A4, and also it's a substrate for PGP. And we'll talk later about whether you need both of those at the same time or just either one alone can cause the interaction. There's some questions about that. But it also can cause, apparently, additive myopathy when it's given with other myotoxic drugs. The issue there is it usually is present in higher concentrations. There's usually some other reason that causes the colchicine to be levels to be elevated. And then if combined with other things like statins, it can result in myopathies. Theoretically, drugs that affect renal function could also increase the risk of toxicity. Next slide. As you know, there are a lot of 3A4 inhibitors, and most of the drugs you see on this list inhibit, not all, but most of these inhibit both 3A4 and PGP. And as a result, clearly have the ability to increase colchicine levels. Next slide. Okay, now we'll turn it over to Lorenzo to talk about his role in this colchicine drug interaction issue.
Okay, thank you so much. I'm speaking from Atlanta, Georgia. So I'm going to show you some of our findings. Next slide, please. So this is a research that we conducted last year and we published in Drug Safety. And as Dr. Hansen said already, clarithromycin interacts with colchicine, increasing its oral viability and decreasing hepatic metabolism. And as a result, there is a decrease in the clearance of colchicine. So, and that could produce like some life-threatening adverse drug reactions. So the aim of this study was to examine the evidence and clinical implications of concomitant use of colchicine and clarithromycin. So we collect information for evidence already published in case reports, and also we use the dashboard of FAIRS database to see reports in that database related with colchicine and clarithromycin. So next slide, please. So we're going to start with the results from the analysis from FAIRS database using the dashboard. So as you can see here, we collected information from 58 report cases. And this is interesting, and it's expecting to that 53% of them were between 65 and 85 years old. 28 of them, they were female. And then it's not surprising also that the use of the colchicine. So 36 of these individuals, they received colchicine to treat gout, 3 of them to treat familial Mediterranean fever, 3 of them Behçet syndrome, and 1 of them amyloidosis. Unfortunately, reviewing this dashboard of the FAIRS database, we found that for those 58 reported cases, 30 individuals die. And of course, we cannot assess that it was because of the interaction, but then that was the final outcome that it was reported in that database. And the other thing that is interesting related with that is 22 individuals received combination of colchicine and clarithromycin to treat pneumonia, for example. And 3 others, they received the combination to treat infection for Helicobacter pylori. So that's an interesting thing to keep in mind. So next slide. And when we take a look about the signs and symptoms reported in the first. So the first one that show up, it was diarrhea. It was more frequently. It is kind of related with the what do we know about this interaction and the colchicine toxicity specifically. And also, but also we have another adverse event that it could be moderate or severe, like a pancytopenia or bone marrow failure or rhabdomyolysis. So it's really important to pay attention to this interaction because the consequences that it can be really serious for patients. Next slide. So what happened with the other portion of this article? So we also review cases reported in literature. And this case report of colchicine clarithromycin co-administration were searched using PubMed and based in Web of Science. And we consider a timeline between January 2005 and November 2019. And individuals' reports were reviewed to identify consequences of concomitant exposure and paying special attention on the dose that they receive, the days of onset, the symptoms, and the evidence of renal disease and time to resolution of symptoms. As you can see here, we collect information for 20 reports. So 35% of those individuals were between 65 and 85 years old. 9 of them were female. 12 of them, they suffer of familial Mediterranean fever. 6 of them for gout, which is the more like a frequent indication for colchicine. Unfortunately, again, from those 20 reports, 4 of them, they die, and 1 of them, it was female. So as you can tell also, the onset of toxicity, it was, in most of those cases, it was within 5 days. And the range was between 1 and 14 days. And the average dose also that we see for colchicine was 1.5 milligrams per day. But 2 individuals received less than that dose, around 1 milligram per day. So also it's interesting that in those 20 reports, 8 of them, they mentioned some renal dysfunction. So next slide, please. So what were the signs and symptoms for those 20 individuals? So again, the most common, it was diarrhea, 75%, but then followed by myalgia, abdominal pain, muscle weakness, and vomiting. So it was a kind of, they followed kind of the same trend really with the findings in first database. Next slide, please. But we didn't stop there. So that was included in the article that I was just showing you. So we are, the next 2 slides, they're gonna show you some preliminary analysis and results from reports submitted to the first database too from 2004 and 2020. And then we were looking for signal of reported adverse event with the use of, in this case, colchicine and clarithromycin. As you can see there, so we are calculating the reporting odds ratio. So the thing that we did is the drug event pairs that we construct for this, in this case, colchicine and clarithromycin, we combine all of them. Each drug we combine, and then we look for the associated outcome. So we generate 2x2 tables to calculate the reporting odds ratio. And the reporting odds ratio shows the odds of certain event occurring with the combination in this case, compared with the odds of the same event occurring with all the other products contained in the database. So as you can see here, the reporting odds ratio are really high. So showing you a strong signal for the occurrence in this case of rhabdomyolysis, agranulocytosis, and acute renal failure when we look for this combination of colchicine and clarithromycin. So Next slides. And then we look also for what happened with colchicine and CYP3A4 inhibitors, and then we can see the same strong signal. So that's interesting, and that's the thing that we are working already. And as I said, these were preliminary results, and we're going to dive more into it in order to provide more results related with the strength of the signal contained in the first database. So next slide. And that's back to Dr. Hanston.
Okay, thank you very much, Lorenzo. Really interesting data that you came up with there. So let's go to the next slide. Now we'll get into some of the details of some of the cases and some of the other reports. This is just one illustrative case of colchicine and clarithromycin that Lorenzo was just talking about. Here was a 67-year-old man who was doing fine on his colchicine, but then they started him on clarithromycin. And notice how quickly this can happen. As you remember, Lorenzo said average of about 5 days, but anywhere from 1 to 14 days. And after only 4 days, he's developed severe symptoms and went on to develop pancytopenia, multi-organ failure, which again, then eliminates the ability to get rid of the colchicine, and it was fatal in his case. There are about 100 not quite, but a little under 100 case reports of colchicine in the published literature, colchicine interacting with other drugs. Okay, next slide. And often in these, this is one particular case, and often in these cases, it's rather complicated. And in this case, it was impossible to figure out exactly which precipitant drug was the primary culprit, probably clarithromycin, but there were other culprits involved. All three of those drugs, atorvastatin, verapamil, and clarithromycin, increased colchicine serum concentrations. But then there were a lot of other interactions amongst the various drugs that all pointed to higher colchicine serum concentrations. So in this case, clarithromycin was probably the primary culprit, but there were ancillary culprits that were also involved. Okay, next slide. And here's a rather disturbing study. Most of these people had renal impairment to start with, 116 people on colchicine and clarithromycin. And in 44 of these patients, the overlap of the two drugs was more than 2 days. And you see the fatal toxicity from, and this was colchicine toxicity, 18% of those people with pre-existing renal impairment who got colchicine and clarithromycin with an overlap of greater than 2 days had a fatal outcome. So this is, and usually from organ failure and or pancytopenia. So this is a rather disturbing look at what can happen when you combine those two drugs. Next slide. We also have pharmacokinetic data that fits exactly with the case studies and the case reports. Here was a kinetic study in 23 healthy subjects given a single dose of colchicine with and without 7 days of clarithromycin. And notice the difference here. The mean increase in the middle there was 212% in the AUC of colchicine, but one person had a rather small 89% increase, although even that would be enough to cause toxicity in some people. But notice in red on the right, the person with the largest effect had an 852% increase. That's very likely to cause toxicity. And the problem with this is that these are healthy subjects. Imagine the variability if you looked at actual patients who are on various other drugs, who have various levels of renal impairment, who have various other diseases, et cetera. So the variability you see here in these healthy subjects is almost certainly larger in the general patient population. Next slide. So here, from several articles, looking at, most of them came from one article, but there's a few extras from other articles. Looking at the percent change in the AUC for colchicine, in purple, you see usually about, most of these studies, there were somewhere between 20 and 24 subjects. So the subject with the smallest change in purple, looking at cyclosporin, 76%, and then the subject with the largest change, 502%. So this is just looking at the variability from, and again, healthy subjects who are more homogeneous than the patient population. Clarithromycin, a huge variability. And look at ritonavir in the middle there, 54% the subject with the smallest change and 924% the subject with the largest change. We'll talk about ritonavir in a minute, but the other thing I'd like to point out to you is diltiazem down there. Notice that, and it can cause colchicine toxicity. There's no question about that. But notice that one subject had a 30% decrease in colchicine serum concentrations. And the problem is you can't tell ahead of time. In all of these cases, there's no way to tell ahead of time whether your patient is going to be minus 30 or plus 339. There's no way to tell that in advance. So that's the difficulty we have in dealing with this. Now, the grapefruit juice, you would think it's known to be a 3A4 inhibitor, so why did it basically have no effect? The mean change was basically 0. And we don't know for sure, but it's possible that because grapefruit juice affects primarily 3A4, inhibits primarily 3A4 with not much effect on PGP, it may turn out that just pure 3A4 inhibitors, there aren't very many of them, but perhaps they don't affect it. Voriconazole also is more or less a pure 3A4 inhibitor, voriconazole, and it also doesn't have much effect on colchicine. So that brings up the question, we don't have the answer yet, do we need to have both 3A4 inhibition and PGP inhibition in order to see a significant interaction? So stay tuned on that one. We should know that in a year or two. Next slide. So taking all this information together, what we're proposing is that if you have colchicine and a 3A4/PGP inhibitor, the first question is there renal impairment? And originally we had an actual value in there for GFR or creatinine clearance, but we decided to take it out because we don't really have enough data to know for sure at what level that you really need to say it's contraindicated with colchicine. So what we put a little hedge in that box to the right, insufficient data, but what we do know is that the greater the impairment of renal function, the greater the likelihood of an interaction. For example, people with really low GFRs, like 20, 25 or so, have dramatically increased half-lives for colchicine. So basically, be very careful of giving colchicine with any 3A4 PGP inhibitor if they have renal impairment. The product information does call that a contraindication. But going down to the second diamond down, if they don't have renal impairment, is there a viable alternative to the 3A4 PGP inhibitor? And oftentimes there is. I mean, just take diltiazem, for example. There are a lot of calcium channel blockers that do not have significant effects on 3A4 or PGP. And if they're appropriate, if they have the same effect as diltiazem, or if they would be a suitable alternative, you should use them. Same thing for some of the antibiotics and some of the other agents. So that's the best way to handle it, is just not give the 3A4 PGP inhibitor. But if you have to give it, and that takes us down to the bottom triangle, or it's not really a triangle, it's a diamond. There we go, it's a diamond. Can colchicine be stopped during the 3A4 PGP inhibitor? And many times that's the case, especially if it's going to be, let's say they're going to be on clarithromycin for a week. Most people, you could just stop the colchicine during that therapy and then start it up again after the clarithromycin is gone. But if you can't do that, then as a last resort, and this is the problem, it's not been called a last resort in the past, it's been one of the first ways of handling it, but we view it as a last resort. That little in the lower right there, use the dosage reduction as talked about in the label. But not just that, but you also must monitor for colchicine toxicity. You can't just lower the dose and say we've solved the problem, and sometimes there's a tendency to do that. You have to monitor for colchicine toxicity, and equally important, the third point there, you have to advise the patient what to look for. So it's not just a matter of dose reduction, but also those other two as well. Next slide. Now, we won't go into the details of this, but this is from the product information on the specific manner of reducing the dose. And again, this is just part of the table. There's a lot more that looks just like this. So yes, you may have to do this, but only we're claiming as a last resort. Next slide. How this ended up happening is this was a scheme basically to, a competitive scheme to have this in the labeling so that other generic companies couldn't come out with competing products. There are now competing generic products, so we don't have to worry about that at this point. Next slide. So this is the argument that I propose for why we should not use dose reduction as a first way of handling the problem. Number A there, the blue A, let's just assume that you're really good at dosing colchicine and you have all these patients who are in green, which is a therapeutic colchicine level. That's the A. And this is before you start the PGP or the 3A4 PGP inhibitor. But let's say that you ignore the interaction and you add the 3A4 PGP inhibitor and you don't do anything to the colchicine. Well, you're probably still going to have a lot of people therapeutic because the interaction won't be large enough to cause toxicity. But in red there, you see, no doubt you'll have, and these are hypotheticals, by the way. This is not based on a study. We're just proposing this is likely what will happen based on the data. In red, some people getting toxic, and probably some people will die. We can predict that given the data that we have. Now, C is the prophylactic reduction in colchicine dose. And the problem with that, and the reason why we call it a last resort, is you're almost certainly going to have some people subtherapeutic for colchicine by making those dosage reductions because of the high variability. All of this is predicated on the high variability from one person to another. And let's say that you're successful under C there, that most of the people are in green, they're therapeutic, but you're almost certainly going to have some people still get toxic even though you've lowered the dose. Just because, again, the high variability. So go to D and you say, oh, well, then let's just lower the dose enough so that nobody gets toxic. Well, then you're going to almost certainly have a lot of people subtherapeutic. So this, again, hypothetical based on the data that we know about colchicine, and this is why we're saying that it's probably best to have that be a last resort, the dose reduction. Next slide. So again, I want to emphasize how important patient education is. Early detection is critical. Some of these case reports are just tragic. I mean, I've reviewed all 80 or 90 of them carefully and assessed them for causality. And it's just amazing how often these people were, even after they had serious symptoms, they continued the colchicine and the interacting drug. And it took sometimes a week or two for them to figure out what was going on. And by that time, it sometimes was too late. So the patient needs to know, not a long, complicated list, but just a short list to look out for muscle pain, muscle weakness, and brown or dark urine. Either one of those is fine, but please don't use tea-colored urine, and I'll tell you in a minute why. This historically has been the common way of warning patients, watch out for tea-colored urine. Not very appropriate these days, I don't think. And also any evidence of bone marrow suppression, such as fever or any other sign of infection or unusual bleeding, which could be related to thrombocytopenia. Okay, next slide. And this is why I don't like the warning people about tea-colored urine. If your urine is the color of that tea on the right, yeah, that definitely is not good. And it may mean you have rhabdomyolysis, but any of those three on the left really don't warrant that kind of alarm. Next slide. So just to summarize, what I think is the rational management of colchicine drug interactions is to try to use, especially try to use a non-interacting alternative to the precipitant drug. If you can't get around that, then either use an alternative to colchicine if it's possible, or stop the colchicine while the precipitant drug is being given if it's a short-term use of the precipitant drug, and only as a last resort, lower the dose. And of course, as I've indicated, advise the patient what to look for. Next slide. Now, just to spend the last few slides here looking at COVID-19, using colchicine to treat COVID-19. As I said, there's almost 24 studies around the world. It's funny, so many people in different countries around the world all came to the same conclusion. Wow, maybe colchicine could be good for COVID because of its properties. And especially for the acute respiratory distress syndrome, some of the mediators of that might be, they think are inflammasome and also various interleukins. And colchicine is known to suppress these things. So it seemed logical that it might be useful in treatment of COVID. So the next slide. This is the Corona trial, which is interesting study because they're looking at non-hospitalized patients, people who, before they get very sick. And the dose in this trial, unlike some of the other trials, is probably going to mitigate the likelihood of drug interactions in the vast majority of people. It's still possible, 1 milligram a day for 3 days. It's possible to get into trouble with that. But then it goes to 0.5 milligram a day for 27 days. So I'm not as worried about this, this dosage causing interactions, although it probably would, but rarely. Okay, next slide. And so again, their inclusion criteria are over on the left side there. And now these are outpatients, but they have risk factors. You can see they're high risk because of one or more of those risk factors. They do have some exclusion criteria, which I won't go into all the details of that. But notice in the lower right, no drugs were mentioned one way or another. Now, as I said, because of the dose, the risk is probably not high. Next slide. But most of the other of these almost 24 studies involve use of larger doses of colchicine that could be a problem. In this COVID trial, there's colchicine starting at 2 milligrams and then 1 milligram a day for 14 days. That's clearly enough to cause serious toxicity if another PGP or 3A4 PGP inhibitor is being given concurrently. Now, they did mention lopinavir, ritonavir and say they would lower the dose for that. But there were no other drugs mentioned, at least at the point that they may have changed these things by now. They may have had some difficulties with the drug interactions, but these were the initial descriptions of their trials that were sent to the clearinghouse. Next slide. Okay, this I think we skipped one, but no, I guess we didn't. All right, next slide. This is the Greco-19 trial where they're giving 1 milligram a day, again, clearly enough to cause a significant drug interaction. But notice over on the right there were no drugs mentioned as exclusion criteria. So unless they've changed that as their study progressed, I'm a little bit worried about this kind of procedure resulting in severe drug interactions. Next slide. And here's the COVID-19 pneumonia trial where the patients, they're treating patients that have pneumonia, and again, 1 milligram a day. They did exclude patients on ritonavir or cobicostat, both of which are very potent 3A4 PGP inhibitors, but they did not mention any other drugs. So next slide. Before I talk about this, let me just summarize the COVID trials. Early results from a couple of these trials, preliminary results, suggest that it may be effective. But even if it turns out colchicine is not effective for COVID, there's still an awful lot of people taking the drug. So we still need to be worried about the drug interactions. Okay, the dose of the inhibitor, I just want to make one last comment before I turn it back over to Dan. And that is that some people think, well, if it's a moderate 3A4 inhibitor, we don't have to worry about it. But a good example is fluconazole, where it is a moderate inhibitor according to the FDA. But if you give large doses, it actually can produce a very substantial inhibition of 3A4. So it kind of depends on the dose of the fluconazole. Small doses seem to have, for most drug interactions don't have very much effect. Yet the recommendations consider fluconazole a moderate inhibitor under all conditions. So I guess the take-home point is that if it's fluconazole or diltiazem or verapamil, any of these that are considered moderate, don't assume that the patient is necessarily safe from severe colchicine toxicity. Okay, I think that's my last slide, isn't it, Dan? Guess it is.
Yes, it is. Yeah, thanks. Thanks, Phil. For those of you that are attending the webinar via computer, feel free to, if you have questions, to put them into the Q&A, and we'll try and address those there. As we get to the end of the webinar, we'll also try to address questions via voice as well, if we can. So what I'd like to do is just kind of wrap up our presentation today, talking about some of the labeling issues with these products. And if you're a pharmacist or a physician and you want to determine if you have a potential problem with your with the medication the patient's going to take with their colchicine, if you go to the colchicine label, you're going to find a variety of information depending upon which label you actually look at. You know, we're really concerned about kind of the one-size-fits-all dosage recommendations. So, but they're actually in the label. There's really not much information about alternatives that the patient could take with respect to those medications that cause problems, and that's not surprising given how the indications for medications might change over time. But it doesn't give the prescriber much information about, you know, what do I go to. There is to some degree shades of gray around the definitions of moderate versus strong. You know, certainly the FDA has its definitions, but even a moderate inhibitor in high doses could potentially act like a strong inhibitor. There's really no information about the dose of the inhibitor, kind of gets back to the previous point. And generally, we're kind of frustrated by the fact that they don't really suggest stopping the colchicine. They provide dosing recommendations or dose reduction recommendations. So if you look at the products that are currently on the market today and go to a source like Daily Med, which is a curated set of product labels, you'll find that there are a number of different formulations that are listed in the current set on Daily Med. 5 labels associated with capsule products, 2 solutions, 17 tablets, and 3 containing probenecid, for which there's no real statements about drug interactions. And if you look across the different columns here, 3A4 inhibitors, PGP inhibitors, that they make a statement about HMG-CoA inhibitors and fibrates, and then things that they say it may be safe with. You know, there's huge variability across the labels. So one of the challenges we have is in terms of managing this particular drug interaction with colchicine is, you know, pulling out the right information. And what we think is the current labeling system for these medications doesn't really reflect the heterogeneity of the data that we have. So I'll just select 3 different ones: the tablets, the solution, and the capsules. I'm sorry, the capsules, and then at the end of the tablets. So let's just kind of take a look at a deep dive at some of the label information that's presented by these different manufacturers. Okay. So this is the capsule. So they do specifically mention some 3A4 inhibitors, such as clarithromycin, ketoconazole, grapefruit juice, erythromycin, verapamil, etc. They do talk about P-glycoprotein, and they do mention, in general terms, HMG-CoA reductase inhibitors and fibrates. They actually go into discussing some drug interaction studies where they've given pharmacokinetically studies, I should say, where they give doses of potentially interacting medications and look to changes. So that's that was the capsules. The tablets, The information is very similar to the capsules. The information is very specific to certain medications, but doesn't necessarily list them all, uses the et cetera. Okay. On the other hand, when we get to some of the branded products, there is a table, as Dr. Hanston showed earlier, table 1, that shows dose reduction recommendations based upon estimated GFR, glomerular filtration rate. And I won't show you table 1 again. Table 4 is fairly small. So here's their drug interaction section, and talks about generally issues with drug interaction. And then this is the table 4 that they provide. So it's a relatively small list of medications that might interact with colchicine and clinical comments about that. All of these are appropriate, but it certainly doesn't give us a sense that there might be other drugs that are problematic as well. And then finally, one of the tables, and the table numbering varies depending upon which label you look at, but essentially the information is the same. They actually give us the percent change in Cmax and area under the curve for colchicine concentrations from baseline, so the change in concentrations. And as Phil mentioned, like diltiazem, 40%, 44% to a 93% increase. I'm sorry, a negative 46% to 318% increase in the AUC with diltiazem. So certainly in some patients, you may not see a change. In others, you're going to see a very large change in the area under the curve, or Cmax, excuse me, and area under the curve. So in conclusion, and we appreciate you joining our webinar today and look forward to the Q&A here in just a minute. We know that colchicine toxicity can be lethal, and our analysis of the Food and Drug Administration's adverse event reporting system suggests that there have been a number of deaths associated with this, with the interactions associated with colchicine. Obviously, we need to worry primarily about the 3A4 inhibitors and PGP inhibitors that are classified as strong, and they can certainly result in increased levels of colchicine. But we also don't have really good data about moderate inhibitors, and we have to be concerned about wide patient variability in response to these. We do know that patients with decreased renal function should be at higher risk of harm. Unfortunately, as Dr. Hanston mentioned earlier, we really don't know. Is that 50 mils per minute? Is it 30 mils per minute? Or is it some other value? Quite frankly, we just don't have really good evidence that would allow us to come out and have a strong decision point when you might want to consider looking at the dose. And then finally, a lot of variability in how the evidence is listed in the product labeling, which can cause concern if that's the source of information when people are making decisions. And therefore, we would encourage organizations and entities, individuals, to look at more than one product label before making a final decision involving a drug interaction with colchicine. With that, we'll thank you for attending. We have a few minutes before the top of the hour, and we'll go ahead and keep the webinar going for just a few minutes and see if there's any questions. There has been one question that was posted in the Q&A, and responses given to that, but if people have others, we'll be happy to try to answer those. All right, well, I'm not seeing any questions come across. Just a reminder for our organization, our research study, I should say, drug-drug interaction, cds.org, and it's hyphenated between drug-drug interaction and CDS. We have a list of resources, including the content of today's webinar will be placed up there so people can view it afterwards, as well as resources for other drug interactions that are of interest. We're also talking to healthcare organizations about the ability to modify their systems to show which drug interactions are important and which ones may not be important to prescribers and other healthcare practitioners. If you're interested in talking to us in a structured interview situation, we'd love to hear from you. You can email us at info@ddicds.org, and we'll set up a time to speak with you. If you're currently modifying your health system with respect to particular drug interactions, we'd like to hear from you as well. We'd like to know what you're doing and how you're doing it, and if it, especially if it relates to some of the drug interactions that we've talked about in our webinars. Look forward to hearing more about what you're doing in the field, so that we can make sure that our support for you is helping you address this particular issue of drug interaction warnings. So with that, I don't see any questions. Maybe I'm just not looking at the right screen, but just confirm with our host. Sheila, is there anything else coming across?
I don't see any other questions.
Okay. Well, thank you again for attending, and best wishes in all your endeavors. Have a good day.